for
biology.
Okay, guys, welcome to Let's Get to the Marks, going through an AQA GCSE Combined Trilogy higher paper for biology. This is the paper one. There's 6 questions. You've got to do it in an hour and 15 minutes. What I suggest is you grab a scrap bit of paper, grab a pen, get your calculator, and then pause the video at the start of each question, have a go, then unpause and check off your answers. And if you can't be bothered to write it down, you can just check them in your head and then unpause to watch the answers. But make it active. Have a go. Question 1. Bacteria can cause a variety of diseases in humans. 1.1. What are 2 similarities between a bacterial cell and an animal cell? And it's worth 2 marks. Tick 2 boxes. Always pay attention to the words in bold. So pause, have a go. All right, going through the answers. So, bacterial cell and an animal cell. Well, all cells have a cell membrane and not a cell wall because a bacterial cell does, but an animal doesn't. Both have a nucleus. No. A bacterial cell has no nucleus. Its DNA lies in the cytoplasm. Both have cytoplasm. Yes. All cells have cytoplasm and both have plasmids. No. So our 2 answers are both have a cell membrane and both have a cytoplasm. Let's move on. Salmonella, food poisoning, is caused by bacteria in food. Give 1 symptom of salmonella food poisoning. Do not refer to vomiting or diarrhea in your answer. All right, pause and have a go. Okay. There's a whole bunch of answers you can put here. And my favorite guess for a symptom for any illness is fever, because often you will get a high temperature when you're ill, part of an immune response. And this one is really for salmonella or digestive disorders. You've got abdominal pain. You'll probably get the mark for saying stomach pain or pain in the intestines. But yeah, abdominal pain. You could say high temperature or sweating because they're symptoms of fever as well. They're allowed, but those are any one of those 2 will get you the mark. Now, if you wrote 3 things, they're going to take your first answer. So always put your best answer first. If it's only name 1 thing, always put your best answer first. Although really, you could say just put 1 answer. 1.3, what is the name of the first antibiotic developed? So pause, have a go. Okay, the answer is penicillin. And that was discovered by a scientist called Alexander Fleming, and they sometimes ask that in exams, but not this one. He says, a child with severe bacterial infection was given a course of antibiotics. Figure 1 shows how the concentration of live bacteria in the child's body changed when taking the course of antibiotics. Of course, a course means they were given a series of pills. Concentration of live bacteria in the body in units. AU just means they haven't given you the moles per decimeter or whatever. So they're the strength of the bacteria or the number. So we've got 1, 2, 3, 4. That's the concentration of bacteria. They started the antibiotics here on day 0. Look, the bacteria was still increasing, but then after day 1, coming down through to day 3 and 4, the bacteria number in the body really decreased until it went to 0 by day 5. We're going to be asked some questions about that graph. So let's get into it. 1.5, the concentration of live bacteria in the body continued to increase after starting the antibiotics. So that's a bit unusual, isn't it? You just started to take some medicine to kill the bacteria, but for the first half a day, the bacteria are still increasing, or for the whole first day, you're still getting more bacteria. Why is that? Any sensible suggestion will do here. And kind of like it's going to take a while. It takes time for the antibiotics to work. But what does that mean? They have to be absorbed into the blood. You swallow the pills, but they don't instantly work. They've got to go to your stomach. They've got to be digested. They've then got to travel in your blood to the right places and kill the bacteria. So they're going to take some time. And there's a whole bunch of answers you could give here that would be sensible. There's a time delay before the antibiotics get to the bacteria. It takes time to travel through the body, etc. Time to be absorbed by the blood, as I've gone for. So anything sensible will get you to mark. After 3 days of taking the antibiotic, the child felt better. There was still bacteria in the child's body. Why did the child feel better? Tick one box, pause the video, have a go. Bacteria had become immune to the antibiotic. The child had become resistant to the bacteria. There were fewer toxins in the body than at day zero. So, why did the child feel better? We have to ask ourselves what was making the child feel ill in the first place. Why do bacteria make you feel ill? And bacteria produce a poison that damages your cells called toxins. So, the reason the child felt ill was because there was less toxin in the body. And that's the reason the child felt ill. Watch out for these ones which say things like bacteria become immune. That's impossible. You can only be immune to something if you contain white blood cells, and bacteria are just a single cell. They don't have any blood. They don't have white blood cells. So bacteria can't be immune to anything. Bacteria can become resistant. So they throw these words in to try and baffle you. But yeah, the answer is the third one here. Okay, 1.6. Suggest why doctors do not give antibiotics to patients with minor infections. So, one of the dangers of giving out too many antibiotics by doctors is we can create something called bacterial resistance. And this is when the bacteria um, develop resistance to certain antibiotics. And that can be quite dangerous. So, they shouldn't give out antibiotics if you only have a little infection. They should just try and let your white blood cells, which we call your immune system, fight it off. So the answer here is because they might become resistant. That's how I'm going to word it and get the mark. You could also say something along the lines of it may develop a resistant strain. So lots and lots of to reduce the development of resistant strains. So anything along those lines. Let's go. We're on 1.7, folks. Figure 2 shows the blood viewed under using a microscope. So here's figure 2, and you can see we've got some blood cells. This one's a little bit confusing and tricky, this question, so it often confuses people. A vaccine will stimulate the production of antibodies. Which part of the blood makes the antibodies or produces the antibodies? Tick one box. Pause the video. Have a go. All right. So, what have we got here? If we're identifying these, which bit produces the antibodies? It's actually B, and B is the white blood cells. They have a very large nucleus, the ones that produce antibodies, and you can see them there. Okay. Which part of the blood in figure 2 starts the clotting process? So, which of these is responsible for if you have a cut on your arm, for you the blood clotting and you getting a scab? And the answer is D. So I'm going to run through what these are because these look confusing. A lot of people will think that A is the white blood cells, and that will make them make a mistake here and tick the box A. And I can't blame you, because what they've done here is they've colored in the center of these circles white, so they look like white blood cells. So that's very, very annoying by the exam board. They've done it to trick you. These are actually red blood cells, and they have a kind of donut shape, look like little donuts. And yeah, it's called a biconcave disc. So they're actually the red blood cells. White blood cells are larger and have a nucleus. So that was the reason why you should pick these as white blood cells. C is the plasma, the yellow liquid that the components of the blood are dissolved in. And D are the platelets that heal wounds and clot the blood. So our answers here for these two are B, produces the antibodies, and the platelets were D, tiny bits of broken cell that clot the blood. So how did you do out of 9? Check your answers. Let's move on. Bada bada bing. This question is about cell division. Question 2. Write the biological structures from the box in the correct order of size. Worth 1 mark. Pause the video. Have a go. All right. What we're going to go through now is smallest to largest. So genes are found inside the nucleus. So definitely genes are smallest. They're simply a little string of code, but actually, chromosome is another name for DNA, and that is also found inside the nucleus. So, chromosome um, is where you find DNA, and a chromosome and DNA are found in the nucleus, and genes are tiny sections of DNA, so they're a small part of a chromosome. So, the next answer is nucleus and then cell. So, see how you did. You have to get all of them right to get the correct mark here. Okay. 2.2 we're coming to, but we've got a little figure to look through here. Figure 3 shows how a fertilized egg can produce specialized cells. So we've got a fertilized egg cell here. What does that mean? That means that a sperm and egg have come together to make that fertilized egg cell. It then divides into an embryo, and then we have a process called A, where it splits into nerve cells, can become muscle cells or red blood cells. So they're probably going to ask us, what is process A? And of course, name process A for 2.2. Okay, hopefully you had a go at that. The answer is process A has several names. You can call it differentiation. It's always a difficult one to spell. Specialization, because it's essentially when cells, these embryo cells, specialize or differentiate into different types of cells. So there we go. Specialization or differentiation, both difficult to spell. How many cell divisions are needed to form a 16 cell embryo from the original fertilized egg? So this is just a quick maths one for you. So pause, have a go. Well, you're really looking at one cell splitting into two cells, and then each of those cells is going to split into two cells. And if we keep going on and on, how long, how many divisions before we get to 16 cells, guys? You should be able to work this out. And then they're going to divide again, and we're going to double. So really, you're doubling again. You could just do this in your calculator. I've been a bit long on the old drawing here, but you can see now I'm going to get to 16. So how many divisions total? So don't worry about these. We just call this one division, two division, three division, four divisions. So the cells have divided four times or multiplied four times. So the answer is four divisions, and that'll get you a mark. 2.4. In humans, a fertilized egg cell contains 23 pairs of chromosomes. How many chromosomes will there be in each of the embryo cells? So, if the fertilized egg contains 23 chromosomes, how many will be in the embryo? Well, when you think about it, this contained 23, and this was the egg contained 23. They joined together in a process called fertilization to make a fertilized egg, which had 46 chromosomes, or known as we have in the question, 23 pairs. And then by mitosis, this divides into a ball of cells, but each one is identical to the fertilized egg. So each one has 23 pairs. So it will be 23 pairs. Of course, you could have said 46, but you're not allowed to say 23 because that would be half the amount that they would have. Okay. 23 pairs or 46 is the correct answer. 2.5 represents a cell cycle for a human embryonic cell. Got a bit of a cold at the moment, so if you hear like if you hear me like wheezing away, that's why. So figure 4 represents the cell cycle for a human embryonic cell. Figure 4, we got the start of the cell cycle here, and we call this stage 1. Then we've got stage 2 is mitosis, and then stage 3. Describe one change in a cell that occurs during each of the stages of the cell cycle. So why don't you pick up your pen, get a bit of paper, and see if you can write down one thing that happens in stage 1 and then stage 2, which is mitosis, and then stage 3. All right, I'm going to go through the answers. So hopefully you've unpaused. Let's go. Stage 1, there's a whole bunch of things you could say. DNA doubles. You don't have to say doubles. You could say replicates. Um, Cell grows in size. So it actually gets bigger in stage 1. We could all say more protein made. Now, more cell structures, things like mitochondria, um, cytoplasm, cell membrane, everything like the cell structures have to be increased because if you're going to split and divide at some point in your life, you need to double everything inside you. So you could just give an example of that and you could say, oh, it had to grow more ribosomes or something like that. But any of those would get you the mark. And then in mitosis, you can literally say any of the things that happen in mitosis. So chromosomes, do you remember what shape they form? They form X shapes, line up in middle of cell, um, go to each end of cell. So really mitosis is all about the chromosomes. So there's a load of things they do. You only have to say one of these. I've said three things. So if you just say chromosomes form an X, that's fine. Chromosomes get fatter, shorter, thicker, become visible. That's fine. They line up in the middle of the cell. That's fine. They go to each end of the cell. That's fine. So any of those would get you the mark. Now for stage three, this is called cytokinesis. Cyto meaning cytoplasm, kinesis meaning like kinetic energy. They move apart. So cyt I can't spell cytoplasm splits, or you could say cell divides into two new cells. And here we go. Right, I'm on 2.6. Cell division is important in the growth of multicellular organisms. 2.6, figure 5 shows how the mean height of boys and girls from birth to age 18 shows the mean height of boys and girls from birth to age 18. Sometimes I don't think I can read. Right, so here's how the height, and we've got age, and we've got boys and girls, how it changes up to 18. So we can see here, boys and girls, pretty similar, their height, all the way up to, in fact, boys seem to be always taller in terms of the mean height, up to about age 11. And from there, the girls take over until age 14. So between 11 and 14, the mean height of girls is bigger. And then boys go back in front at 14 and stay bigger up till the end of our graph, which only goes up to 18. So we're going to now have a look. 6 marker. What? Compare the growth of boys with the growth of girls. Use data from figure 5 in your answer. Beautiful one, this one, because nice and easy, but you just got to be clear and concise. They're testing you on how you can read a graph. So what we want to do here, folks, I'm going to write my answers up here because I can explain as I go through the graph. So my first point here would be to say that up until the age 11, boys are always taller than girls. So mean height of boys is larger than girls up to age 11. I could also say the opposite. The mean height of girls is lower than boys up to the age of 11. And then between 11 and 14, um, between 11 and 14, uh, girls have a higher mean height. That doesn't mean that every girl will be taller than every boy between ages 11 and 14. It means if you did, and I've called girls grills, it means if you did an average, the average height is bigger. I've called girls grills. So we don't want to call girls grills. So that scored me 2 marks straight away. There's loads of points we can make. And then over here from 14 to 16, we can say boys have a higher mean height. And then we can also say the data only goes up to 18. Yes, darling. You want to play what? Excuse me. Okay, some other general statements we can make. We can say that girls when do they stop growing? Looking at this, when does the line level? The line appears to level at age sort of 13 to 14. Girls stop growing at ages 13 to 14, whereas boys stop growing. They grow for a little bit longer. It looks like boys stop growing about age 17. So you're just using the graph. You could be a little bit more accurate if like you draw some lines on the graph as well. The examiner will like that as well because it shows that you're justifying your answers. So I'm saying boys stop growing at 17, maybe it's more like 18. And I've said that girls stop growing at like 13 or 14, but they might still be, that's still kind of curving there. I might have been better off for saying 14 girls stop growing. But there we go. Like you're just showing, if you leave those lines on the graph, the examiner sees what you're doing. And this would have already got me 6 points. But there are some more complex answers you can give here. You can sort of pick any line and state the difference in the height of boys and girls at that age. So for instance, let's go to 12. If I bring the line up here and then bring it across for girls, we can see that girls are like 144 centimeters, whereas if I bring the line up to find out where the boys are about yeah, it would help if I could draw a line straight. The boys are about whatever that is. What's that, 152? So about 6 centimeters taller. So I could say at age 12, boys are 6 centimeters taller than girls. So you can do some of that if you like. If you like to get challenging and put some of that stuff in, you can say boys keep growing for longer. Look, boys are still getting taller there where girls have stopped growing. So we can say, we can just put a general statement. Boys keep growing for longer than girls. All of that stuff. There's loads and loads of points there, but you can see I've done enough. But there are tons of other points that you can make. You can do comparisons at any of these ages between the heights and quote the heights. And here we go. You can talk about where they're the same height. So I've missed out where they're the same height. Look here, same height here, same height. So what's that? That's 14. So at age 14, boys and girls, the mean height is the same. And this looks like at age 11 or just before 11, 10 and 8 months or something, the mean height of boys and girls is exactly the same. So yeah, there are other points you can make, and that is going to get you big, fat 6 marks. We've got one more part to this question too. 2.7, give one way that cell division by mitosis is important in fully grown animals. So what they're saying here is don't say, oh, they need it for growth because an animal has already fully grown. So why do we still need to, does an adult like myself still need to do mitosis? Well, of course, your hair grows, new skin. When you scratch yourself, you get new layers of skin. So we call that repair. For repair of tissues and cells.
If you push the button.
Would you like it?
Like.
Amylase is an enzyme that digests starch. Which organs in the human digestive system produce amylase? Tick one box. Liver, small intestine, large intestine, salivary glands, stomach and liver, salivary glands, pancreas and small intestine, stomach, pancreas and large intestine. And the answer here, guys, is the salivary glands, the pancreas and the small intestine. And in fact, a little note to make, pancreas and small intestine make all the digestive enzymes you learn about at GCSE. So they make lipase, protease, and amylase, which is a carbohydrate. So pancreas and small intestine, if you have to remember one thing that makes enzyme, go for pancreas or small intestine. That's just a little tip-top tip for lazy people that don't want to do all their revision. Right. A student investigated the effect of pH on the activity of amylase. This is the method used. Prepare amylase solution at pH 5, mix the amylase solution with starch in a boiling tube, remove a drop of amylase starch mixture every 30 seconds and test it for the presence of starch. Record the time when all the starch has been digested. Repeat steps 1 to 4 using amylase solution prepared at pH 6 and then pH 7 and then pH 8. 3.2. What was the independent variable? So top tip, when you're looking for the independent variable and you have a method, it's normally in the last step. So you will normally find the independent variable in the final step of a method. You can see here, the independent variable is what they change. And what are they changing? They're changing the pH. So pH, pH of amylase or pH of solution. Yeah, let's just put pH of solution. Let me change that to black because I've been writing in pink for a while now. There we go. pH of solution. So that is the independent variable. They're changing the pH. Of course, you may be asked, what is the dependent variable? What are they measuring? Well, they're investigating the effect of the activity on the amylase, and they're doing that by testing for the presence of starch. So what they're actually measuring is whether starch is there or not with the iodine, but they're not going to ask us that. So describe how the student would know all the starch have been digested. So they're adding iodine. Do you remember what iodine does with starch? So iodine goes blue-black with starch. So if there was no starch in there, the iodine wouldn't go blue-black. The iodine would stay normal iodine color. And I don't know if you know what the color of iodine is. It's like brown or orange. So we'd say the iodine would stay brown or orange. So it wouldn't go blue-black. You might even get the mark for just saying the iodine won't go blue-black. And that is what the answer would be for that. So I think you get the mark as well. If you can't remember that it's orange, if you just say iodine does not go blue or black, then you will also get that mark. Cool. 3.4 shows the student's results. Figure 6, time taken to digest all the starch in minutes. What was the optimum pH for the amylase? So this is how long it took to digest the starch. So for instance, at pH 5, it took the starch 10 minutes to digest the amylase. Is that optimum? Normally, this is another trick question. The examiners are being bummers to you, and I'm going to draw on in pink. Normally, when we look at the optimum, we look for the top and we go, oh, look, that is the top. That's the optimum. But this time, that means it's working really slowly. They've reversed the graph. And actually, the optimum pH is down here because the optimum pH is the pH where it takes the quickest time for the enzyme to break down the starch. And that is going to be over here. At, the time taken was like 1 minute and something, but that is pH 6.5, 6.8. So what was the optimum pH for the amylase? The answer is 6.8. Let's check. Is this multiple choice or no? It's not multiple choice. We just write the answer in here. So 6.8 is the optimum pH, the best pH for breaking down the starch. Now down here, more graphs. Don't you just love a graph? A scientist did a different investigation. This is the method used. 1, prepare amylase solution at the optimum pH. Mix the amylase solution with starch in a boiling tube. Measure the concentration of sugar every 10 seconds for 2 minutes. So here's the time. Here's the concentration of sugar. We can see that over time, the concentration of sugar is going up. How much time did it take for the amylase to digest all the starch? So here, how do we know all the starch has been broken down? And hopefully you'll realize that when starch gets broken down, we have a little bit of a reaction. So starch gets broken down into glucose, which is a sugar. It actually gets broken into maltose as well. But for the purposes of this, that's also a sugar. But for the purposes of this, so why did I put iodine? Lost the plot. So what I want to do here is put amylase. So amylase is the enzyme that's turning the starch into glucose. When has it finished its job? It's finished its job when that line straightens, no more sugar's being made. It means all the starch has run out, so no more sugar is being made. And that happens at around 80 seconds. You may want to say like 85 seconds, 82 seconds, but I'm just going to say 80 secondos and get myself a nice fat mark there. It's actually, they'll give you anywhere between 80 and 84. The exam board say that's 82 is perfect, but anywhere between 80 and 84, and they'll give you that mark. So lovely bubbly. Right. Let's zing down. Now we have determine the rate of sugar production per minute at 40 seconds. 4 fat marks. What they want to do here, this is looking at a tangent. So when you get a curve on a graph, did they say 40 seconds? I think they said 40 seconds. They said 40 seconds. I just had to double check. Right, let's go down. Bish. What you do here, each of these is going to get you a mark. If the examiner sees you do this and draw a line on the graph, there's 1 mark for you. Okay. What you're looking to do here is draw a tangent. So you're going to draw a circle or a dot or a plot at 40 seconds. The next thing you need to do is draw a line to smooth out that curve like this. Now that would not get you a mark. That line's not touching. And also it's not equal on both sides. So what you want to try and get with your line, and this is pretty tricky, you want to try and get it so that by your eye, by the examiner's eye, it's roughly equal the spacing between that line and the curve on each side. And I'm going to say that's roughly a good job. But obviously on a computer, it's a little bit harder. Try and draw the line so it's touching the axes on both sides. That's how I do it. You don't have to do that. And then what you want to do is make a little triangle anywhere. And this is going to get you another mark by doing this. So I'm making a little triangle here and I'm drawing my triangle there. Okay, that is the crustiest triangle in the world of triangles. I'm going to redo that because that wasn't very straight. All right, let's try this a little bit straighter. There you go. Now, notice what I've tried to do is pick at least one number to make it easier for myself, like the 4 there, okay? And then we're going to label this Y is the Y-axis, always going up, and X. And I've done my tangent now by drawing this line straight here. So I'm going to get a mark for drawing that dot there, a mark for drawing that straight line. And now I want to measure Y and X, and I want to do Y over X equals rate. So I want to divide Y by X. It's really the change in Y. I can use this triangle as change in Y over change in X equals the rate. So I just need to work out what these numbers are here. So this is 4 for Y, but Y started over here at 2.5, 2.6, 2.7. So Y is 2.4 over 2.7. So I'm going to write that here. 4 take away 2.7. That is Y. You could just write 1.3. And then over here, this is 20. For X, you go down to the X axis. And then over here, bosh, bash, 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 bash. What is that? That's 50, 54, 58. No, that's not 50, 48. Sorry. So 20 to 48. And let's see what that gives us. I should have done 48 minus 20. There we go. So we want to do 1.3 divided by 28. Smash that in the calculator, and that is your final lovely jubbly answer. 1.3 divided by 28 equals... I need to find my calc. Da, 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 da. I'll have an answer in yeah, so I got like 0.046, and when you look on the mark scheme, I actually wouldn't get a mark for that. They want the answer to be 0.042. So I can't really make this any larger in my crappy eyesight. What's that? 4 2.6, 2.7. That looks pretty good. The 1.3 is decent, but it's probably my 20 to 48 is a bit rubbish, and it's probably more like 44. So it's probably more like what is that? That's 20. Oh, I'm so dumb. I'm so thick. Check this out, peeps. This is why it's best to be a little bit more accurate. So that's obviously not 48. What a stupid Wally I am. That is 50 there. So if that's 50, that's going to be 58. Ah, dear, oh dear, oh dear. So that should have been 58. So there we go. I lost a mark. I'd have still got 3 out of 4 marks for doing y over x. You get another mark. And then just because I got the wrong answer, but because of my reading on the graph, would only cost me 1 mark. But really, we are looking at 1.3 apparently divided by something like, no, that's not 58. What is that? If that's 50, these are going up in twos. That's 54. Oh my gosh, I can't. Yeah, I'm going to give up. I'm struggling here, peeps. I'm struggling to read a graph. It's all going pear-shaped. Jeez Louise. So 1.3, what's that? 54? Yeah, 1.3 over 24. Da, 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 da. I can't. I've fallen apart. I've fallen apart. 54 minus 20. 1.3 divided by 34. Oh my good gosh. That's much better. That's within the range. Oh yes. 0.038. So there we go. If only I'd read my graph correctly, 0.038, and that falls within the range. They're giving you on the mark scheme something like that to that. And so obviously there's going to be variation in how people draw the triangle and measure it and that type of thing. But that was just bad reading of the graph. You see there, that is 54. And I read that first as 48. How can it be 48 when 50 is there? And then I read it as 54. Can't be 54. Sorry, I read it as 58. Can't be 58. It's not over here by 60. So it's actually over 54. All of that cost me a mark, but in the exam, we go, hey ho, there we go. So 0.038 will get me the correct full 4 fat marks. That was a bit long, wasn't it? Nearly had a heart attack doing that question. Okay, 3.7. Explain how the structure of enzyme molecules is related to the effect of pH on the activity of amylase. So explain how the structure of enzyme molecules is related to the effect of pH on amylase. So what we want to do here is talk about enzymes. So enzymes are proteins. They have an active site. So hopefully I'm going to draw some diagrams as well so you guys can see. Right, that's an enzyme. It's got an active site. The active site has a specific pH that is complementary to the substrate. Basically, it fits. Now, that's some fat word, isn't it? Complementary to the substrate. What we want to say is, if you prefer, this might be better. It fits the substrate like a lock and a key. You can say the induced fit model, like a hand in glove, if you like, or like a hand in a glove, if you know about the induced fit model. That's a more updated model. Induced fit model. So that's a more updated model. So like this, here's the substrate and it goes in there. There's the enzyme, there's the substrate. What we can say, once they join together like that, only one type of substrate, e.g. starch, can fit into one type of enzyme's active site, e.g. amylase. So really, I've already said that with the lock and key, that there's specificity, that the substrate can only fit in there. At the optimum pH, the fit is perfect. However, outside the optimum range of pH, the enzyme denatures. Now, do you know what denatures means? We normally talk about denaturing in terms of temperature, kinetic energy, destroying the active site. But actually, the active site's shape can go all messed up, even at a different pH. It's because of the hydrogen ions. pH is a measure of the hydrogen ion concentration, and different hydrogen ion concentrations affect the bonds that hold together the active site. They disrupt it. The active site loses its shape. So the active site changes shape and no longer fits the substrate like a lock and key. So we can tell from all of this that it's absolutely vital you have the correct pH so it can digest the starch. If it no longer fits the substrate, this means it can no longer digest the starch into sugar. So the active site and the enzyme is useless. It's basically dead at this point. It no longer can digest the starch into sugar, so it doesn't have any purpose at that point. And of course, if all your enzymes got denatured, you'd die. That would be a bad thing. But enzymes get denatured every day, folks, and we make new enzymes all the time as well, so don't panic. Right, question 4. Photosynthesis is an important chemical reaction in plants. Why is light needed for photosynthesis? So what is light? It provides the energy. Remember, photosynthesis is an endothermic reaction. So it provides the energy or activation energy. Gets the reaction going, right? What is the correct equation for photosynthesis? Pause that video and have a go. So what do plants take in? Well, they take in carbon dioxide and water. So this one looks kind of good. We can definitely get rid of that. They don't take in sugar. They take in carbon dioxide and water. But what is that? They give out oxygen, but that is not the formula for sugar. So we can get rid of that. Okay. They take in carbon dioxide and water, liking it. They make glucose and oxygen, liking it. That is the correct one. Of course, here it's got them taking in oxygen. It's not the equation for photosynthesis. Plants do actually allow oxygen to diffuse in when they need to respire. If they're not getting enough oxygen made by photosynthesis, then they can take oxygen in. Okay, 4.3. A student investigated the effect of different colors of light on the rate of photosynthesis at room temperature. The student used pondweed in water. A piece of pondweed was placed in red light, then blue light, then green light. Each color light was the same intensity. Describe how the student should make accurate measurements to obtain valid results for the rate of photosynthesis. So essentially what they've done here, they've got some pondweed. What an incredible drawing. And what I would do actually is I'd put it in a conical flask, but yeah. So they've probably got some pondweed like this. Now, you could say count the bubbles. Pondweed is going to produce bubbles of oxygen gas. We fill that with water, but do you know what? Better than counting the bubbles, bubbles can be different sizes, is to use this piece of equipment. Do you know what this piece of equipment's called? Should do. And if you don't, this is your chance to write it down. This is a gas syringe. It accurately measures the volume of gas released. So that would be important. But we need to control, if we're changing the light in color of light, we need to control all the limiting factors. Do you remember your 3 limiting factors of photosynthesis? So what I would say is, yeah, let's say a little place pond. You probably don't need any of these to get the marks, but this just gets you the feel. Place pondweed in a conical flask with water. Connect to a gas syringe. Woo, to measure the volume of oxygen released. Of course, we're going to need a stopwatch because we need to make sure that is the most incredible stopwatch. We need to make sure that we can measure the time the oxygen's released. Measure the time. for a set volume of oxygen to be released, we need to keep certain things the same. What are the three limiting factors? We've got temperature. So keep the temperature the same using a water bath. So you need to know that. That always comes up, of course, because if you increase the temperature, you're giving the plant more kinetic energy and it's going to react faster. Then you don't know if it's the green light or the red light or the blue light. So we're going to use a water bath to keep the temperature the same. You don't need a diagram, by the way. That's just me trying to help explain it. We also want to keep the same amount of sodium, hydrogen, I can't spell, sodium hydrogen carbonate. Okay, why do we put sodium hydrogen carbonate? Sodium hydrogen carbonate is basically baking powder, and we place this in the water to release carbon dioxide. You wouldn't put it in the water bath, you'd actually put it in here. But yeah, that's going to release carbon dioxide and allow the plant to photosynthesize. So they've all got the same amount of CO2. And the next thing is keep light intensity the same by using a ruler to keep distance of lamp from plant same. Okay, so what I'm saying by that is we want to have our lamp. Now we're going to change the filter so we get red light, blue light, green light, but we need to also keep the distance the same. It's not fair if I move the lamp closer with green light, then of course it's going to look like green light has a better effect. If I move the lamp closer with red light, yes, it's going to look like red light has a better effect. We want to find out, is it the red, is it the blue, or is it the green that gives off the most oxygen bubbles in the same time? So maybe you say, how much oxygen can you give off in a minute? And you start your timer and you start counting how many, or you don't need to count the oxygen bubbles. You're measuring the volume of oxygen gas in the syringe after 1 minute. So we need to keep the temperature the same amount of carbon dioxide to the plant, and the same light intensity. And so this would get you all the marks. So, scientists investigate the effect of different wavelengths of light on the rate of photosynthesis. The wavelength of light determines the color of light. Figure 8 shows the student's results. Figure 9 shows the scientist's results. Rate of photosynthesis, red, blue, green. So what we've got here, red light, blue light seems to be really good at doing photosynthesis. Green light, not so good. And this is the students' results. Let's look at the scientists' results. Oh, they did a line graph. Rate of photosynthesis, and they did different wavelengths of light. Why are the results for the two investigations presented differently? So it's because the independent variable is different. So we've still got the same rate of photosynthesis, but we've got a different independent variable. Here they've got the independent variable as red, blue, and green light, which is a discrete or a discontinuous variable. And down here, they've used the wavelength of light instead of the color of the light. And the wavelength is on a continuous spectrum. We call that a continuous variable. So that is the reason why they presented the stuff differently. Nice. So what we want to say is student has used you might know it as categoric variable, color of light. You can call that discontinuous or a discrete variable. Those are two other names for it. The scientist has used a continuous variable. And whenever we use a continuous variable, we do a line graph and we do bar charts for categoric. So that's the answers there and there. Ish bash bosh. The student suggests the range in the wavelength of green light. Use figure 8 and figure 9, range in the wavelength of green light from to that. Okay, so what you're doing here, you're doing a little comparison between the bar chart and here. So you're saying, okay, green light had an absolutely rubbish rate of photosynthesis. I think the rate of photosynthesis was, not me and graphs again, somewhere between 0 and 1. Let's call it half, 0.5. So in arbitrary units. So I want to go 0.5 here and come along. What is the wavelength of the green light? It's going to be somewhere, where's 0.5, like there. It's going to be somewhere like here Yeah, it's this kind of section around here. That is where the green light is. You can see it's really rubbish there. The amount of photosynthesis is pretty rubbish, and that matches where was the photosynthesis rubbish on this graph. It was rubbish with the green light. So we know that the green light is the one that is not producing a lot of oxygen, and therefore it's somewhere in this graph. So I would go for something around, I don't know, they're going to accept anywhere on the example between like 500 and I don't know, I could play it nice and safe, like 500 to 550. So that's what I would do. I go 500 to 550. Keep it nice and safe, and that will get you the mark. Of course, the exam board allow a whole range, so they'll allow you anywhere between like 475 and like 577. But let's just double check because I'm not being very accurate there. So I'm going to take my time and just double check this. So let's have a little look, folks. What did we say? It was about 0. What did we say? It was about 5? What was the arbitrary units in photosynthesis up here? So yeah, okay. Yeah. So I reckon mm, yeah, maybe. Yeah. So you're looking at the lowest part of here, I reckon around there. So that's where our green light is. Okay, cool. I might actually go. Does it really matter? 480 to 520 sounds good to me. Okay, that is the wavelength of green light according to my incredible calculations. Question 5. This question is about tumors. Describe the similarities and difference between a benign and a malignant tumor. So, similarities, both are a ball of cells that are growing rapidly um, due to uncontrolled mitosis or cell division. Okay, so when some of your genes mutate, um, this can happen. You can get cells that will just start growing rapidly with uncontrolled mitosis, which is cell division. And so what we can say is both result from changes in DNA. Do you know the word for a change in the DNA? If you do, you can put that fancy word down, and it is mutation. And that is a change in a DNA. And you need to know that. You can also call that a change in a gene, specifically changing a gene. So these are similarities. I'm going on similarities. I think that's how you spell it. So these are my similarities. I've done 3. That should be enough. Also, the cells are abnormal. So the cells are not normal. And you could get up to 3 marks for this. You won't get 4 because they want some differences. So that if you haven't done any differences, they're not going to give you all 4 marks. So let's do some differences. So malignant, oh days, malignant tumors are cancers. So malignant tumors are cancers. They grow and invade other tissues and organs. So they grow and invade other tissues and organs. Um, Malignant grow faster. So they're even faster growing. Um, I don't need to keep writing malignant, but there we go. Malignant. Oh my good gosh, malignant. Um, Not only do they invade other tissues and organs and they grow faster, and we call them cancers, but malignant tumors can spread. So you can have lung cancer and then it can spread to your liver, spread to your spine, spread to your kidneys. So malignant tumors can spread to other organs or parts of the body. Cool. Ah, and they travel in the blood, whereas benign don't. There we go. That's enough differences. Benign tumors are also encapsulated in a membrane, which is why they can't invade other cells and tissues and things. But all of these are loads of differences, and loads of similarities. You only need 4 of those guys. Okay, figure 10 shows data about skin cancer in males and females for different age groups in the UK. It shows the number of new cases of skin cancer in 1 year, the number of people with skin cancer per 100,000 population in 1 year. Here we've got figure 10. This is one of those difficult graphs. You can see the number of new cases of skin cancer is up here. Then on the other axis, the opposite y-axis, we have the number of people with skin cancer per 100,000 population. We then have new male cases and new female cases represented by the bars. What that means is if you have to do anything with the bars of data, you read the bars over here from the left, this data on the left. With the dashed line, which represents the number of males with skin cancer, and the solid black line, the number of females with skin cancer, you read that data from the right here. Okay, so anywhere you go up to the black line and you read off of here. With the bars, you read off of the left. Okay, let's check it out. 5.2, there are no new cases of skin cancer diagnosed in people younger than 15 years of age. Explain why. So we come up here and we look here and we say, okay, there's no new cases of people below the age of 15 with skin cancer. Why? Why do you think? Well, it takes time for the cells to mutate. It takes time to develop skin cancer. They may not have been exposed to as much sunlight or ultraviolet radiation. So they have not been exposed to enough sunlight yet. We could say it takes time for cancer to develop. We could say it takes time for DNA to mutate. So there's lots and lots of reasons we could give here, but any 2 of these reasons would be good enough. So I want to say less time exposed to radiation. What type of radiation? That would be ionizing radiation. You'll know from physics, alpha, beta, gamma, the stuff that actually can cause mutations to cells. So they've had less exposure to X-rays and to ionizing radiation. Diddly-dee, diddly-da. Oh, less exposure to carcinogens, cigarette smoke, burnt food, all different types of things, food wrapped in plastics. They've just had less exposure. They've been on the earth less. They've had less chance to get their DNA damaged and for it to mutate and for them to develop cancer. So the best answers here are for it takes time for DNA to mutate, less time to expose to radiation, less exposure to carcinogens, less exposure to sunlight or ultraviolet radiation. Those 3 are the best to take your 2 marks from. Okay. Give 2 conclusions about the number of new cases of skin cancer, 2 marks. Use figure 10. So I'm going to answer this one up by figure 10. And what we've got to do with this one is zing up to the actual graph and answer it up here. Now it tells us 2 conclusions about the new cases of skin cancer. So any 2 statements about new cases of skin cancer, and we could just say, let's have a little look. If we look here, it looks like with new cases of skin cancer, let's have a look. We're looking at the white and the grey bars. So you could really pick on, ooh. In fact, look at the women's bars are the white ones. They're always taller than the men's, all the way up to the age of 59, and then the men take over. So what we can say here is, whoops. Up to age 59, the females have, what's that? Oh, jeez, Louise. There we go, sorted it out. Up to the age of 59, females have a higher number of new cases of skin cancer. So that's one thing that we can say. When you're drawing conclusions, you want to say definite things that are 100% factually correct. And we can factually say that these white bars are taller than the grey bars. So females have more new cases of skin cancer up to the age of 59, because after that, males have higher. So what we can say now, we can say the opposite and get the 2nd mark by saying males have higher number of new cases after the age of 59. Look, little cheat code. We said one, let's say the other. We can even say when the number of, are there anywhere the number of new cases are equal? No, they're always different between the male and female. There's none where they're, no, that one's close. Close, close, close. Which year has the greatest number of new cases? So we could say 65 to 69 as the greatest number of male cases of skin cancer and the greatest number of female. So yeah, I would say that's an important point to point out. Age, what was that? 65 to 69 has the greatest number of male and female new cases. So that is the age range for both males and females where you get the most new cases of skin cancer. Now, I'm running out of room here, so I'm going to stop there. You only needed two, but there are loads of other factual things you can say here that would pick you up the marks. And remember, you're looking at the bars, not the lines for this question. Right, let's zing to 5.4. The data for the number of people with skin cancer is given per 100,000. So just why the data is not given as the total number of people. Well, the reason why they do it per 100,000 is because there might be a different amount of women and men living in the country. So it's not really a fair comparison. But by doing it per 100,000, you're basically lowering the population and saying, if we just look at 100,000 and compare it like that, it's easier to compare. So easier to compare. You might just get them up for saying easier to compare, but best to say because the population of males and females is different. So they're always going to be slightly different. So it's much easier to compare populations by doing it per 100,000. Okay, let's zing down to 5.5. Describe two trends shown in figure 10. They've repeated figure 10 up here. Use only the data for the number of people with skin cancer per 100,000. So this time we are only looking at the dashed line and the black line, and we are describing two trends with the dashed line or the black line. So, well, one trend is the number of cases, number of people with skin cancer per 100,000 increases for both men and women over time, over the age in years. Let's have a look at some other trends. Number of males with skin cancer increases with age. That is going up with age. Number of females with skin cancer increases with age. The number of males with skin cancer increases more rapidly after age 55 to 59, whereas the number of females with skin cancer is more of a steady increase. So there's a difference there. Look at the males' skin cancer is accelerating after age 59. We could say that the females are higher than males up till age 59. After age 59, the males are higher. So you just pick any two of these trends that you can spot and write them down here. So the two I'm going to go for are number of males per 100,000 increases with age. So that one was fairly simple. The line goes up. As they get older, more people get skin cancer. Now, we could have said that for females as well, but that's the same trend. So they probably won't give you, like, it sounds like an easy one. I'll just say the number of males increases with skin cancer with age. And I'd say the number of females increases with skin cancer per age. But that's the same trend. So they might not give us that. So we might have to come with something a little bit more nuanced. Something like number of females increases with age, but at a steady rate. And I could say number of males per, I should have said per 100,000 for the females as well. Number of males per 100,000 rapidly increases after age 59. So after 59, 55 to 59, we see that steep increase in the number of males developing skin cancer per 100,000. Anywho, there's a nice basic one. And then these are slightly more nuanced, but there we go. Let's zing on. So, 5.6, the estimated population of males aged 80 to 84 was 694,000. Calculate the number of males aged 80 to 84 with skin cancer in that year. Give your answer to 3 sig. What we need to do, it says use figure 10. Always pay attention to that. You can't calculate it without using figure 10. So you come up to figure 10 here. We zoom in a tad and we say they told us it was age 80 to 84 and it was males. Males are the dashed line. And so we zing across to the right. Remember, we read the lines from the right, we read the bars from the left. So that looks to me like we need to calculate how much each square is worth. So we go like that. There is 20 and there are 5 squares. So I'm going to do 20 divided by 5. That equals 4. Each square here is worth 4. So that's 104, 108, 112, 116. So my magic number is 116. And that is what I'm going to take back down and do that calculation with. So we now zing back down. Where's the calci? There it is. 3 siggy. They want it. So there were 116. Okay. What did that tell us? 116 males per 100,000 had skin cancer. So 116 males per 100,000 had skin cancer. There's a total of 694,000 males. How many altogether have skin cancer? Well, we want to do 694,000. I'm going to divide that by 100,000. That gives me, not V, that gives me 6.94. You don't need that multiple because you're just going to then times that by 116. And why do we do that? Well, if 100,000 has 116, then I want to find out how many 100,000s are in 694,000. There's 6.94 100,000s in there. So 6.94 lots of 116. And when you do that, you're going to get something along the lines of this in your calculator. It told us 3 sig. So if you leave that as your answer, you will only score 2 marks. One for working out the 116, one for doing the calculation. Whoopsie there. And the last mark is for the answer to 3 sig. So the actual answer to 3 sig is 805. If this had been 805.74, then that would round up to 806. But it's not. It's 805.04. So 805 is your final 3 sig. And here we go. Right, we're on to the last question. Let's zing down quickly. So this question is about the heart. Why is the heart described as an organ? So it is made of different tissues. So the heart contains things like elastic, muscle, fat. So made of different tissues, and they are performing the same job. But I can say function or role might be a more funky word to use. So there we go. Remember, a tissue is made of similar cells, but we were asked about an organ. So 6.2, valves in the heart keep blood flowing through the heart in one direction. Shows the heart valve labeled here. Explain the effects on a person if the valve there developed a leak. So this would be the same for really any valve developing a leak, but essentially let's zing, oh my good gosh, let's zing up a little. Got some crashing and banging next door, but don't worry about that. So what happens is the blood returns from the lungs. It's full of lovely oxygen. It comes up here, zings around the back, down the aorta and off to the body to deliver that oxygen. If there's a leak, the blood will fall backwards and we'll get less blood going to the body. So what we want to say is there will be backflow of blood. Right. What does the backflow of blood mean? That means less blood to cells in body. If you've got less blood, what does the blood carry? So less oxygen to cells. Now, if there's less oxygen to the cells, what is it that those cells can't do? Why do they need oxygen? So less respiration. Boom. If there's less respiration, specifically what type of respiration, because maybe on the mark scheme, if they get a bit funky, they'll say only accept aerobic respiration. So less aerobic respiration. If we can't do as much respiration, what do we make less of? less energy. And that means the person's going to feel tired, sore, fatigued. They might collapse, all those kind of things. You can see we've scored loads of marks here. I'm going to go over to this side here to write some more points. So if you can't do aerobic respiration, what type of respiration can you do without oxygen? So we could say more anaerobic respiration. The body's going to desperately try and make energy to stay alive. So we'll do more anaerobic respiration. That is going to mean a buildup of lactic acid. And if you wanted to write down how that's going to make you feel, it makes you feel sore, pain, tiredness. So all of those things. Anyway, having less energy is going to make you feel tired. And of course, you could slip into a coma and die with these kind of things. So there we go. 4 lovely marks. You just need some of those things there. But I would always say this is the easiest one where you go less blood, less oxygen, less aerobic respiration, less energy. And that happens a lot with the heart. Any issues with the heart tend to lead to those 4 things there. So you can kind of apply that to lots and lots of faults, holes in the heart or chambers that are not connected in the correct way, all that kind of thing. Right, final part of the question seems to be 6.3. Faulty heart valves can be replaced using biological and mechanical valves. Oh my gosh, look at all this info. The faulty valve is replaced during an operation. Biological valves are from animals or humans, allow blood to flow through them normally, wear out, stiffen over time, need to be replaced. Mechanical valves are made of synthetic materials, can cause blood clots on the surface of the valve, require anti-clotting drugs to be taken for the rest of the life, can last a very long time. A young woman enjoys extreme sports and would like to start a family. The woman needs her valve replacing. Describe the advantages and disadvantage of this young woman of having a biological valve instead of a mechanical valve. So what are the advantages to a biological valve? Well, biological valves do not cause blood clots. So biological tissues are very, very smooth, smoother than the smoothest mirror, and they won't cause blood clots. Whereas mechanical valves can cause blood clots. So the downside to that is she would need to take anti-clotting, it tells us in the information, medicine, um, medicine. Um, the danger of extreme sports is that a cut could become very serious from blood loss. If you can't clot your blood because you're taking these anti-clotting medicines which stop your blood clotting in order for you not to die of a heart attack from a blood clot, then that means if you get a cut while doing skiing or an extreme sport, motorbike riding, you could just bleed to death and lose enough blood that your pressure drops and you die. So that would be a bad situation. So that's a downside to mechanical valves. However, mechanical valves last a long time. Um, so mechanical valves last a long time, which is very good. And also biological valves um, can be rejected. What does that mean? That is when the antigens on the biological valve from the donor do not match your cells. Antigens and your white blood cells think they're foreign and attack them and destroy the new organ that's supposed to save your life. So biological valves can be rejected. She would need to take immunosuppressants for the rest of her life, which makes her more susceptible to getting colds, flus, COVID, that type of thing. So that's a downside to that as well. Also, there may be a waiting list for donors. So what that means is there might be a lot of young children in front of you. She may have to wait a long time before she receives an organ, and it might be hard for them to find an organ that matches her biology perfectly. So that's one of the other issues as well. And you just really need any 4 of these things to get yourselves all 4 marks. So any 4 of these would get you all 4 marks. Uh, let's zing on. That's the end of the questions. Oh, lovely, lovely, lovely. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.